panel. So I encourage all of the attendees to put panel questions into the Q&A. But we have also some pre-prepared questions, which I will start with. So first, and we can just take a minute for each panel member to respond, at least initially. Based on all the results that we've seen, so there's some very interesting similarities across our 4 studies and also some differences across the 4 studies. What would you say are some overarching take-home conclusions that we can glean for T-cell responses to SARS-CoV-2?
A quick introduction, sorry. Can all
sure.
the panelists please turn on their videos for this section so that we can see your faces? We didn't do that for the previous session because we are fearing that there's some bandwidth issues if we are showing the slides and the faces. So this will be a trial and error attempt. And also for the chat, if you have any questions that you want to ask, please use the question and answer tool of the webinar system. We cannot respond to raised hands. Thank you.
Oh, thank you, JP. So, shall I restate the question?
So I think the question was about what are some broad takeaway things, is that right?
Yeah, I mean, it's very interesting. I noted that some of the studies had some shared results, some overlapping conclusions, and then there were some also differences. But maybe now we have some take-home conclusions.
I'll offer a trivial observation, which is that T cells are clearly important, or at least play an active role. And we've seen very strong signatures shared both at the phenotypic levels and the antigen specificity levels and the receptor levels. I think the causal role is what we'd all love to understand next.
May I can add, I thought it was quite interesting, and I think this is something that's, I'm still more in your data, Jonathan, is the dynamics that I think both Donietta and what we've seen is that there's different types of dynamics and different populations responding at different times. And I think this is something that keeps coming back, but maybe we don't fully understand. So I actually have a question about that, but maybe I can let other people answer first.
Yeah, I think so too. What I think was also interesting in our analyses was to see that there's some pre-existing immunity, to see in these pre-pandemic samples that we actually collected, or let's say these clones from these patients could be found in these pre-pandemic samples that we collected, I think, over the last 6 years maybe. And I think the distribution of these clones in these different cohorts, I don't think that's random, because that really mirrors what we're also seeing right now, right? We're seeing that the risk groups are actually individuals over the age of 60 and people with other diseases like cancer, for example.
Yeah.
I mean, from our side, probably the biggest take home is finding these completely new subsets. Cytotoxic CTLs, cytotoxic TFH cells. There's a lot to learn about these subsets. And the relation to these recent studies that are showing people with fatal infections don't mount a good germinal center responses makes us wonder whether these cytotoxic TFH cells are important. And the other piece is in terms of the clonal expansion that we find, these CTLs seems to have huge clonal expansion. The clone sizes are 3 or 4 clones in some patients that dominate the repertoire. Surprisingly, we don't find a lot of public TCRs. We do see a few, but in the 40-odd patients that we looked at, bear in mind this is straight analysis without any expansion. It's a 6-hour stimulation condition, so we're not expanding the cells. So that may be a factor.
So in your experiments, Vijay, remind me the timing when the samples were collected. Can you say anything about how the subsets you observed may relate to the day 15 versus the later expansion peaks that were observed in the first two studies?
So it's a very, very, very interesting thing. These cytotoxic TFH responses are clearly seen in samples where we collected early on in the illnesses.
Okay.
So, 10 days to 20 days is when you start seeing these huge cytotoxic responses. So that's why people probably haven't seen these in convalescent samples or, other viral infections in the past. In an acute infection, 10 to 15 days, we see these huge responses. So clearly time is a factor here.
Mm-hmm.
So can I ask a question actually about Jonathan's, you showed this graph where you said that in the people that recovered and didn't have major symptoms, the TCR counts were much larger. What day was this, or was this an overall statement? Or
that was looking at the cohort level. So one of the cohorts from Bloodworks Northwest was exclusively convalescent patients. So I'm not sure the exact dates from those, but the enrollment criteria was that they had cleared virus by that point.
Okay, so they can actually be comparing people at different time points. I mean, different cohorts can be taken different.
Yeah, the cohorts are all over the place from very early time of PCR diagnosis to up to a couple months post clearance of their last symptoms. So more than that since infection.
Cool, thanks. Another sort of thought I had was about cross-reactivity and specificity, and that, as Vijay was saying, they don't see so much of it, and others see a lot, and obviously it depends very much on the HLA type. But this may, sort of regardless of COVID and saving the world and all that, this may be an interesting place to look at how cross-reactivity is restricted by HLA types and how it goes beyond that, if at all, right? So I guess you guys have the data to look at that.
Yeah, it's an interesting question, and of course, for pre-existing immunity, the ideal data sets would be to get blood before exposure, which is definitely the hardest cohort to get your hands on. But certainly, as we start identifying signatures of responses against these shared epitopes and start looking across individuals, both historic controls, but also then exposed uninfected, mild asymptomatic, I think it'll be really interesting. And actually then pairing that VJ with the functional data that you guys are generating, I think it'll be really interesting to understand how those are different both in healthy controls, but then also after exposure to SARS-CoV-2.
Okay, so if there's no more discussion on that, then I'll move to the next question. Are there assays that you might suggest for diagnosing and or treating severe versus milder cases of COVID-19, and especially for predicting severe versus mild outcomes early on in the disease process, such as following T-cell subsets or clonal expansions, or even for monitoring for viral exposure or return to work?
So I just have one comment on that. One is, there's a couple of interesting papers. There's this issue of breathlessness and long-term symptoms and fibrosis that is seen in people with COVID. There's a set of papers from a couple of groups that have shown that these cytotoxic CTLs are strongly associated with certain fibrotic diseases. Mm-hmm. So the question then is, you were seeing these fibrotic changes in these people pretty early on, they're persisting. What's driving these reactions? Mm-hmm. If there's precedent on CTLs being associated with other fibrotic diseases, one could speculate, if you mount a profound CTL response, are these cells contributing to fibrosis? Mm-hmm. So, we're looking at some studies where we're examining these CTLs more long-term to see whether they are linked to people with fibrotic outcomes.
Mm-hmm. Well, I do know that the cytokine profile a particular cell type produces at a site can dictate whether you get, sort of tissue damage versus tissue healing. What do you know about the subsets that you have found in terms of whether their cytokine profiles tend more towards the wound repair versus damage phenotypes?
It's a difficult one to speculate. These CTLs and other subsets, they're making all the right cytokines. They're linked to protection in some instances. They are linked to pathogenesis in some instances. Particularly CTLs, they make a lot of dendritic cell recruiting cytokines, myeloid cell recruiting cytokines, but it's difficult to tease apart whether they are playing a protective role or pathogenic role. So the associations is what we can draw right now. But just that in other contexts, the fibrotic diseases, these CTLs have been linked to these processes. Pretty convincing data from Shiv Pillai's group in a couple of fibrotic diseases. So finding this in a few patients, then the question is, we are following up these patients long-term to see whether these patients have greater fibrotic consequences compared to people who don't have these subsets.
Very interesting.
So can you remind us when you saw them peak time-wise? The CTLs?
Well, so the cytotoxic TFH cells are seen early on in the illness, whereas the conventional, the helper CTLs, they're seen at different time points. So the follicular cytotoxic is something we see early on. That's a different phenotype.
And specifically, did just day-wise, early on, meaning around the...
So early, so this is between 10 and 20 days from the onset of symptoms. And in people with a severe disease, that's the main thing. We see this pattern mainly in people with severe disease. And when you look at their transcriptome and predict the upstream regulators, interferon signaling components come up. So whether the profound interferon response that you see early on infection and severe infection, whether that's skewing a CTL TFH response is what we're looking at.
Very interesting.
In terms of the diagnosis question, I think this is a really, using T cells to diagnose is a new field, right? And I think it's pretty intriguing. What I'm excited about it from our perspective is that on the one hand, we know that T cell responses persist much longer in at least SARS-1 infections, and I think a lot of the early data for SARS-2 is consistent with that. But more importantly, we can, with any of these high throughput broad sequencing approaches, we can measure responses across the entire proteome. And so just from a diagnosis of past infection, we expect that these sorts of approaches will be more robust to, for example, viral variation, different types of responses that people have, and eventually will allow us to start teasing apart what's the difference, what is the actual prognostic role of these. I think as we get more into prognosis, the science is it becomes more and more of a scientific question at this point. Like we don't know the roles of these things. There's a lot of open questions. We certainly don't know the causality in these things. I think it'll be very exciting to look at that, but there's just a lot of science to be done there. And in terms of the question about immunity, I think it's the same question. We can very confidently say whether somebody has an existing T cell response to a virus, whether that's indicative of immunity is very hard to say at this point.
So have you thought about, your control cohort has like 3,500 people now. Have you thought of checking whether any of them got COVID since you sampled them?
Yeah, it's an interesting question. We've limited these to pre-2020, which of course doesn't exclusively rule that out. I don't believe we have the ability to call any of them back, unfortunately, just because of the protocols. That would be an ideal scenario, of course, but I don't believe we have the ability to do
And prognosis, there are two separate questions, right? So one is pretty much who gets really sick, and the other is, why are some people recovering and having long-term consequences? And that seems to be the bigger thing that's emerging. People are having long-term breathlessness and symptoms.
Yeah. Absolutely.
So the CTL that I'm talking about is mainly to look whether that's linked to any long-term fibrotic consequences.
Mm-hmm.
The part, it's difficult to nail any predictions at this stage.
Mm-hmm.
Well, I think one thing is the mild disease that people can cure at home, let's say, and the other thing is the need for hospitalization. And at some point, I think it would be really interesting if we have data, or let's say, like we said, sequence pattern that we find in patients with mild disease that can really predict, or these clones that we found, for example, these recovery-associated clones that can really predict that this person has a high chance of really recovering fast from this disease, or at least with symptoms, with no need for hospitalization. But I think for that we need much more data, and I think the best or most informative cases will be those who actually have mild disease, but from whom, unfortunately, we rarely have samples during their active disease because they're not in the hospital, right? So it's really hard to get these samples at that time point when it's when we can really early on see what's exactly happening during that mild infection.
Mm-hmm. Yes.
To that point, we do have in the public database from ImmuneRace in particular people that self-identified as having COVID, and many of those cases we can go back and verify PCR positivity. But it is a small set, and by definition they're now recovered, not early on. And so this is a place where additional partnerships, we're definitely looking to expand the partnerships towards many of these other studies that have looked at small numbers of asymptomatic and mild cases. But you're right, those are by definition harder to find. and I get worried that we spend too much time looking under the rock of severe hospitalized patients.
We're looking where the light is shining. Yeah. So if no more discussion on that point, maybe we could shift to thinking about vaccination. So, obviously with vaccination, it's very important to think about which peptides are included, for example, which kinds of T cell subsets you steer towards. What thoughts do you guys have based on your results so far that would inform vaccine design?
I'll say that I'm happy that we're even thinking about the role of T cells in vaccine design. I think that's been a long
yeah, it's no longer all about neutralizing antibodies.
It's a maybe disappointingly new science. There's certainly been decades of work in it, but in terms of actually pushing it all the way into clinical use, I think there's a lot that's unknown.
Yeah, it's harder, right? And we have less success there. But it may be what we need in this case, right?
Yeah, and I think there's interesting questions. You can look at it in multiple different ways. What's the role of T cell responses in helping generate the antibody response? A lot of what Vijay was showing, I suspect, is directly linked with that. Then there's a separate role of what is the role of CD8 CTL responses, or maybe even the CD4 CDL responses in protective immunity. Completely unknown. And the third question is, if they are important, how do you even generate that in a vaccine? Which of course has been the source of a lot of research in the cancer community, but not so much in infectious disease outside of HIV that I'm aware of.
Mm-hmm. Well, and what about having vaccine components that are designed to avoid particular T cell subsets as well, that might be important in light of, I think, some of Vijay's results?
So Lindsay, the way, there are two aspects, right? So what we looked at is in the acute phase, in the first, 20, 30, 40 days. So we're bringing back these patients and reassessing these responses in these patients. This is like, 2 months, 3 months apart from the onset of illness.
Mm-hmm.
And I think that's where I'll probably be able to learn what is the stable protective immunity that's persisting.
Mm-hmm.
So what I would like to see in a vaccine study is very much to see, what are the protective phenotypes that are seen in acute or mild or severe infections. Do we get these clones or these phenotypes following vaccination? That's one part of the question. But I agree, if the problem is we don't know whether these CTLs are good or bad. At the moment, all we have is an association.
Mm-hmm.
So we can't say if vaccine induces a CTL, that's a bad thing.
Mm-hmm.
We know from the yellow fever vaccines that you can induce a CTL response, and that's associated with good kind of protection. So it's difficult to know whether that holds true in all these infections. So I think it's important to look at, we have the acute kind of phenotype. We need to know what are the nature of T cells when people are fully recovered. What is protective memory responses? Do we use those responses with vaccines?
Right. Yeah.
And it's a question of stability, right? So the big concern is, would we have, long-term memory with these vaccination or even real infections?
Mm-hmm.
We don't know whether we see these T cell responses in these milder cases, 90 days post-infection.
Mm-hmm. Mm-hmm.
So,
yeah.
So I have a question for you, for everybody. Would you worry that there's a possibility of overstimulation? So in the beginning, we heard that a lot of the really, really bad critical cases were due to cytokine storms. So is this something that could happen, triggered by in one way or another?
it's possible, right? But all we have is an association. The cytokine storm could all be myeloid cells, and then you have a proper protective response that's kicked off by the T cells. So the associations at what we have right now, we can't say whether this is driving the process.
Mm-hmm. Thinking back to the first two speakers that presented these two peaks, right? This first clonal expansion peak at day 15 and this later peak, which may or may not be associated with mild cases. It makes me wonder whether in designing a vaccine, perhaps it's necessary to induce both of these types of cells, whether they have different specificities or not, whether they are of different T cell subsets or not.
Yeah, I mean, I think that's a good point, and the idea you put forward about the secondary peak being due to some sort of regulation and repression. I think that that's partly also where my question from a second ago came from. So I think it's really interesting to try and understand the dynamics because this could really play a role.
Yeah, we need to merge all of these studies into one, right? Where we have the longitudinal data on the mild cases and the severe cases and have their HLA information and have this mirror assay all on the same people.
Yes. But I think the beauty of the vaccination studies is, the time you gave it, you're following up these patients, and the outcomes in these patients. So then it'll be very nice to correlate, I find a CTL, are these people developing adverse reactions or not? Mm-hmm. So what we have from COVID patients is this is the range of responses you can see. We don't know what these responses mean. Mm-hmm. What are the consequences of these responses are. And this is something you could easily address in a vaccination study.
Mm-hmm. Yes. Which hopefully, I guess, well, does anyone know? So all the vaccination studies that are currently underway, or at least the ones that are in humans, I guess these are all focused on eliciting antibody responses, or are some of them also looking at T cell responses? Does anyone know?
As far as I know, they're collecting blood for assessing both responses in the couple of vaccine studies that I'm looking at.
Mm-hmm.
The vast majority of them are focused only on surface glycoprotein components.
Yeah.
I mean, they're not inducing CTL responses or T cell responses, but they're focused on getting antibodies.
Yeah. That's the primary endpoints in there. But they're collecting blood for exploratory studies.
Yeah.
Yeah. So, just reading through the last couple of questions here. So I think we've touched on most of these questions that we had pre-prepared. So do you guys have questions you would like to pose, or again, I'll ask for additional questions from the attendees? Some of you, I think, looked at serum antibody in conjunction with your T cells, right? But you didn't present that in today's talks, but I believe I saw that referenced in some of your papers. Is that right?
Yeah.
Do you have anything to say? I know we're going to really focus on B cells tomorrow, but, and I don't know if any of you are presenting your B cell data tomorrow, or if it's a wholly different set of speakers. But can you say something about the coordination between the two, if you observed anything?
Well, to be honest, coordination, I don't have anything of that in mind, but I definitely know that what we saw was a lot of naive B cell responses, which was really in line with what you were saying about the germinal center reaction probably not really working well. So definitely we're on that. We're sequencing more and more repertoires that are coming from whom we have a lot of clinical data, and then we can just try to find, because we also found some B cell clusters that were actually associated with some patients who had a better outcome. And yeah, but we didn't look into the dynamics of the B cells and the T cells during the timeline. So I don't have any of that information.
Yeah, because I was just wondering, given what you observed and Alexandra observed with these 2 peaks of clonal expansion, is there a similar dynamic on the B cell side that might hint at a similar 2-peak dynamic of different clones or anything like that?
We didn't look into the 2 peaks. I mean, we looked at the antibody levels at the time the blood was collected for T cell analysis.
Mm-hmm.
And what we find is subjects who had higher cytotoxic TFH response had lower antibody levels.
Mm-hmm.
As I mentioned, the caveat there is cytotoxic TFH cells are seen early in illness. So time is a confounding factor.
Yeah.
So we're bringing these patients back, and we're starting the analysis where everybody is, say, 90 days post-illness, and now we can see the antibody responses and start relating that to if they had a marked cytotoxic TFH response, are they having lesser antibody levels later on?
Mm-hmm.
Right.
So a couple of more questions have come in on the chat. So one person is asking, I guess this is directed at VJ, how to characterize the new T follicular helper cells, given that their numbers are so low. And same question for the CTL CD4s.
I think, it depends how you would like to do the assay. I mean, right now, single-cell RNA sequencing is a pretty straightforward assay. These assays, you would easily see these population clustering. So the standard way we do is the peptide pool. We stimulate them for a period of time, 6 hours or 24 hours, isolate the cells and phenotype them. You can do phenotyping by fax or by single-cell RNA sequencing. With FACS, certainly staining with granzyme and perforin works well. Granzyme works well compared to others. So if you have a cell, CD4 cell that's expressing granzyme, it's a CD4 CTL. Then comes the question of whether you can separate that into a follicular CTL or a non-follicular CTL. That can be slightly tricky with surface markers. CXCR5 and other follicular markers may help you.
Okay, great, thank you. Well, we have just a couple of minutes left, and so
I have, maybe if I can ask a question, which is more technical. When will the MHC class 2 mirror be available?
It'll be in the next release. I don't know exact date on that, but weeks.
Okay, thanks.
Yeah, so I was going to just ask for closing comments or questions from the panelists, and then also give an opportunity for Pam or other of the organizers to tell us anything we need to know in preparation for tomorrow.
Do the panelists have anything to add? If not, I can just tell you what's going on. Last chance. I'm looking at you. You don't have anything to add. I can see that. It's been excellent today. Thank you, everyone, for attending. I know it's quite late in Europe, and we still have a really strong attendance. That's great. And thank you to Lindsay for moderating, and to all of the speakers and panelists today. It's been a really interesting conversation, and I'm looking forward to the next 2 days where we'll have sort of the same layout tomorrow. We'll have 4 panel speakers, 4 presenters tomorrow, and at the same time. So in the Pacific time zone, we're starting, it's a 3-hour span, it's 8 to 11. And then on the day 3, we'll have 3 different sets of panels. And so if you're interested in knowing more about the full program, it's up on the website. And as I said, we'll start again tomorrow at 8 a.m. Pacific. So that's all I have to add. Thanks, everyone.
Great, thank you. And thank you again to the speakers. Those were very interesting talks. So thank you.
Thank you.
Thank you all.
Cheers.
Bye, everybody.
Bye.